Hello, welcome to the MicrobesFlux website. In this demo, we are going to show you how to use the website to reconstruct genome-scale metabolic models and perform constraint-based modeling. We provide a name for the model, an email address for getting optimization results, and choose the interested species in KEGG. Here in this demo, we use CYN as an example. We click Run, and immediately we will get the genome information of this species. Next, we can get a detailed view of the pathway by clicking Metabolic Pathways on the left. Here on the right, we can inspect each individual reaction, knock out reactions, or make modifications to them. For instance, we can knock out R02569 by checking the KO box on the right and saving it. We can also add user-defined reactions, including biomass composition, inflow, outflow fluxes, and heterologous pathways. For example, we can add 1 ATP to biomass composition. We automatically combine all biomass compositions into one reaction. We can validate compound names by using the Validate Pathway button. Once the pathway is validated, we can add it to the pathway list. We can add inflow reactions to indicate compound transportation from outside to inside the cells. Similarly, we can add outflow reactions to indicate compound transportation from inside to outside the cells. In addition, you can also add heterologous pathways that do not exist in the original genome. Our reconstructed genome-scale metabolic model can be exported to the SBML format and can be visualized. The website will send SBML and pathway map files to the designated email address. After the reconstruction, we can proceed to constraint-based modeling by clicking Optimization on the left panel. We use flux balance analysis, FBA, to determine the flux distribution in the reconstructed model. On the right, we can see three tables. The first one indicates the objective function. We can choose maximizing biomass as our objective function, or define our own objective function by assigning weights to each flux. The second table shows the flux balanced equation automatically constructed by MicrobesFlux. The third table lets us view and modify boundary conditions of each flux. After making all necessary changes to the model, we can submit an FBA job as an optimization problem to our system. We can also submit a DFBA job to simulate the dynamic metabolic behaviors. In this case, we will have to upload a data file that specifies the inflow and outflow fluxes induced earlier. We use cloud computing as the back end to the large-scale optimization. The results will be delivered by email as well. Our MicrobesFlux system is open to everyone and free for academic use. We don't need to register or log in at all to use the features of the website. However, registered users can save their models for later access. Here we will show you how to use our website to reconstruct a toy model and run constraint-based modeling. The complete pathway information of this model can be found in the link below this video. We first choose Toy, a toy example, and click Run. The genome information shows that there are 10 pathways. Next, we go to Metabolic Pathways. The biomass composition has been defined in the toy model. At this point, we specify the inflow and outflow fluxes for the toy model. We select the reaction type to be inflow and type C_GLU as the reactant and C_G6P as the product. This indicates the glucose transport into the cell. We then validate the pathway and add it into the model. Similarly, for outflow, we put C_ACCOA as the reactant and C_ACT as the product. This indicates the acetate transport to the outside of the cell. We then finish the model reconstruction and move to the constraint-based modeling phase. In optimization, we choose the objective function to be maximizing biomass. We also change both the lower bound and the upper bound of inflow 1 to 11.0, and change both the lower bound and the upper bound of outflow 2 to 6.4, and then submit it as an FBA job. We can also perform dynamic flux balance analysis for the toy model. We upload a data file with flux information of inflow 1 and outflow 2 at each time interval, and submit DFBA job. The results, which include genome information, metabolic pathways, generated FBA or DFBA model, and flux distributions, will be sent by email. You can see a sample of the results of the toy model by clicking the link below this video. I hope you enjoy using MicrobesFlux. Thank you.